Assalamualaikum and hi, I'm Nur Hanani, and today I would like to present about detection of bacterial pathogen in aquaculture sample by DNA microarray analysis. For the introduction, rapid, sensitive and specific detection of pathogen intended as a guide for the optimal use of antimicrobial agent. It is important for effective disease control in aquaculture. According to Brian, Loy and Bodrossi in 2006, microarray-based analysis is gaining popularity because it can simultaneously provide many pathogens in a single diagnostic assay. This method is based on the fact that even a single base mismatch between an oligonucleotide probe on the microarray and the target gene sequence can weaken the hybridization signal intensity. Recently, microarray assay have been used for the detection and identification of aquatic pathogens. For the objective is to develop a DNA microarray assay to detect species of pathogen bacteria such as Vibrio cholerae, Vibrio harveyi, Vibrio aeruginolyticus, Vibrio parahemolyticus, Aguilarium, Nocardia cereole, Aeromonas, Hydrophila, and Streptococcus in aquatic animal. So the problem that have been found is member of the genera Vibrio, Nocardia, Aeromonas and Streptococcus, which are ubiquitous in environment, are common contaminant of aquatic product. Some species are opportunistic or less frequently occurring pathogen of human and many other animals can causing considerable economic losses. For microarray method, is all the oligonucleotide were dissolved in double distilled water to a final concentration of 40 m. And then 5 UL of the prepared DNA solution was buffered and mixed with the same volume of DNA spotty buffer. In a 96-hour plate, multiple microarray were printed on open minus slide and glass slide, so that independent microarray were present within each of the 4 wells. Each probe was then deposited at a fixed location within a mass well by using a smart TM48 spot. Each probe was printed at 3 repeated spots within the microarray, and every array also includes 4 quality control probes. After probe spotting, the slides were baked for 1 hour at 30 degrees Celsius is an oven, then processed slides were then rinsed and stored at room temperature in the dark. That they use for detection bacterial pathogens, genomic DNA was extracted with the universal kit for bacterial DNA extraction quantified by 1% agarose gel, and was subsequently used as a template for PCR amplification. The universal primer, pair GYRB forward and GYRB reverse, was used for the amplification of an approximately 1.2 kb fragment of the bacterial GYRB gene. Further, the primer pair 8 forward and 1391 reverse was used for the amplification of an approximately 1.5 kb fragment of the bacterial C60S rRNA gene. PCR was carried out in a 20 UL reaction volume containing 1 times PCR buffer, 2.5 mm MgCl2 200 UL of each deoxynucleoside, 3 phosphate, 500 mm of each primer, 2.5 ULA tag polymerase and 2 ULA DNA template. Amplification was performed in a master cycler personal thermocycler by applying the following 3-step program denaturation for 5 minutes at 94 degrees Celsius 30 cycle. each of 30 seconds at 92 degrees Celsius, 30 seconds at 54 degrees Celsius, and 30 seconds at 73 degrees Celsius. And final extension for 7 minutes at 73 degrees Celsius, the PCR product were electrophoresed on 2% agarose gel stained with ethidium bromide, and visual is under UV light. In this result, show that DNA was extracted from the blood of 21 disease animal and then used for microarray testing of the 21 sample. So 60 sample has been produced a positive result, and only 19 different bacteria were identified. In particular, Aeromonas hydrophila, Fibrio harvey, Fibrio parahemolyticus, Fibrio anguillarium, Fibrio alginolyticus, and Fibrocolaria have been identified at the species level, and Aeromonas species that have been identified at the genus level. The fluorescence signal intensity for healthy and negative hybridization sample were below 200, while positive hybridization sample had exceed 1000. Experimental infection tests show that most of the symptoms caused by this pathogen were similar to those found in natural disease fish. However, to isolate of Portabacterium damsile from Mitis amio and Nibia albiflora can only cause mild symptoms such as skin ulcer and have a lower mortality rate, namely 2 bacteria strains from the Pleucococcus altivelis, 1 from Plicoria crocea, and 1 from Strinus carpio was identified as Aeromonas species, which is it at the genus level. Okay, for the discussion, the disease management and evaluation of culture aquatic animal are major concerns in commercial aquaculture. Rapid identification of pathogens in aquatic animal is important not only to avoid economic losses but also to enable animal health management. Based on the result that obtained using 2 different methods has shown that microarray test has produced accurate result and can identify not only at target bacteria but also unknown bacteria such as Aeromonas species at genus level. The detection sensitivity for Vibrio anguillarium in fish blood is less than 100 colony forming units. Furthermore, the time required for pathogen detection in blood sample from diseased aquatic animal is less than 5 hours, which is more efficient than the PCR culture method. Furthermore, the 16S rRNA gene sequence has been widely used for the characterization of bacterial population, but these genes cannot be used to differentiate closely related fibrous species. Recently, GWRB has been identified as a suitable gene for identifying bacterial species. In this study, we target both the 16S rRNA gene and the GWRB sequence to design a microarray probe for identification of the bacteria.